right? Okay, so in across the world's oceans, looking at just coral reefs alone, there are more than 1 million species that are projected, right? This is not what's known, but what's predicted to be occurring on reefs. And that's around, if you look at multicellular species, just species that have lots of, more than a cell, right? It's multicellular species, including corals, fish, and so on. That's predicted to be about 800,000 species, right? And actually only about 8% to 9% of that projected diversity is known and is named. And that is a relatively low number considering that we have been over three centuries describing species, right? So we are really quite far behind. If you look at species that are named, it's a small proportion of those that are undiscovered and those projected. Of course, this is just multicellular species. We're not even talking about the small things, right? Microeukaryotes, those single-cell organisms, right? Organisms that exist in single cells, maybe existing as symbionts in corals and other things. There are really quite small numbers that are known, but we know for sure that the actual numbers are really much higher. Bacteria is a black box so far. Just looking at symbionts, that means species that are living within reef taxa, reef organisms. There's 74 of these species, you get 20,000 species. So this is a vast underestimate, and you can see that as you collect more and more symbiont or hosts, you get more species, and that increase hasn't been stabilizing. So we're expecting to see a lot more species that are present, these tiny microbes, right? So just looking at taking a step back and looking at marine species generally, right? The prediction is much higher. It's about 2.5 times of the coral reef species, about 2.2 million species that have been predicted. That is over here. That is actually a relatively small number compared to what's on Earth, right? So the ocean really just contains 25% of all Earth species. So although we tend to say that coral reefs and marine environments are very diverse, they are actually not as diverse as terrestrial habitats, in fact, right? But what's important here is to take note that out of all that 2.2 million projected species, actually only about 9% of that has been cataloged. That means they have a name attached to that, right? And you will see also that apart from this predicted number, there's also a standard error. This is basically the variation around the estimate. It is a very huge variation, right? Looking at Earth species and also in marine species. So why are we so uncertain? This is a list of the estimates of marine biodiversity throughout many years, right? Going from the 90s to 2000s to today, right? The most recent one is in 2011, 2012. And you see that there's a vast, quite a big range. And of course, what we are quite certain of is the number of species that are described. And that is really, regardless of what estimate you use, is a small percentage of what we have been projecting for global marine biodiversity. The estimates have landed to around 2.2 million, but it goes up to 5 and sometimes even 10. So why are we so uncertain about marine biodiversity? Right, this is a huge range of numbers, right? And essentially, I think the main reason is that we don't really know what's there yet, right? So majority of species have not been described, and that's why. So if you have most of these described, then we actually can close in on a suitably precise number, but we haven't, right? Majority of these species have not been described. So how do we find these undescribed species, right? The more species we find, I think that is important. The more species we find and we describe and characterize, the more we are certain about biodiversity. And that gives us an idea of what we stand to lose, right, if we don't conserve species. And so there's a lot of implications for conservation, not just at the global level, but at site level and locality level, where we want to have good estimates of the number of species at each site so that you can make conservation decisions. Okay, and so let's go into what we are really missing, in a sense, what are the parts of marine biodiversity that we really don't know much about and where we need to focus our efforts. Okay, so of course, the obvious one is the unexplored deep, right? So this is A good example of the museums initiatives in more recent years, we have been venturing more into the deep sea. In 2018, we, together with our Indonesian colleagues, we organized an expedition to South Java over 14 days. And just within those 14 days, more than 12 new species, and the number is growing by the month. As more are being found and described. New species new to science, new records for Indonesia, right? Lots of specimens collected, right? So this is a good example of, an environment that is unexplored, and every expedition you go to, you will find new species. Of course, further away, you will find even more unexplored areas. This is in the Arctic, right? So you see this accumulation curve where increase the number of samples, you don't get stabilization of the estimate. So these curves are continually increasing, meaning we have not reached some kind of precise estimate of how many species there are. So we need to collect more and more. We need to collect more and more samples and so on to get to that stage. Okay. But I won't be talking about deep sea or the Arctic that much, right? That I think, and that's really not my my expertise as well. Coming back home, right? We just need to look at Singapore's marine environment to get a sense of what we are missing. Okay, we are right at the western edge of the Coral Triangle, the world's most diverse marine region. It is a small marine area. We have about 1,000 square kilometers of total sea area. It's dwindling, of course, or getting smaller as we reclaim from land into sea. But out of this 1,000 square kilometers, only about 25 square kilometers, a very tiny area, are made up of our major coastal ecosystem. So we have in blue, in purple, coral reefs, in red, you have mangroves, and in green, you have the mud flats, and which can have, or sand flats, which can include seagrass habitats on that. So these are very small areas, and these are where most of our species records have been from. Okay, so what are we missing from all these places, right? And of course, the most obvious ones are just the very poorly studied groups, right? And one reason why we don't really have a good sense of marine biodiversity is because the biodiversity in the marine environment is very varied, right? And not just in the number of species, but also in how disparate they are, right? So this is a good example. So if you look at animal phyla, this is the biggest group, right? All the big body plants, the very general body plants we're talking about. So for instance, you have chordates, right? You have Cnidaria and so on. There are 33 animal phyla, and 32 of them occur in the ocean. If you compare that on land, only half of that, right? 12, or more than half, but 12 out of that 33 animal phyla actually live on land. So basically, animals that live on land are quite similar to one another, but animals that live in the ocean are quite different from one another. This is the same if you look at class, like the class level, and if you look at order level, family level, whatever it is, you get much larger numbers in the oceans. That just testament to how varied the organisms are. So when they're so varied, you rarely get taxonomies or biologists that can specialize across these major groups, right? And so that's a problem, right? Very few taxonomies specialize across these major groups. Okay, so if you look at this is a list, kind of not comprehensive, but a list of all the reef species that reef groups that you can find, big groups like you have corals, you have hydrozoans, you have mud dragons and ternaids, and these different groups. You can see that they're all very varied. You seldom have a taxonomist that can study, like I work on corals, but I know nothing about crabs, right? And someone who studies crabs will know very little about corals unless you're studying crabs that are symbiotic on corals. So we tend to specialize a lot, and so when we do that, we neglect certain groups. Like one good example of a very fairly understudied group is bryozoa. These are called moss animals, and just it took us a long time, but we managed to get experts into Singapore, and then we just in a couple of weeks we found 3 new species of bryozoans, and this is one that is just right under our noses. You've been diving a lot, you will see a lot of seaweed, this sargassum seaweed, and on those sargassum seaweed, some of them can have this bryozoans that are encrusting on that seaweed. So these are things that we we always observe seaweed, but we don't actually look at what's encrusting on them, right? So new species that's been present on these groups. Of course, there are the tiny stuff, and I mentioned a little bit of that. So just featuring a bit of dinoflagellates. So these are a group with quite diverse, about over 1,500 species worldwide, and they are generally living in the water column, and they are the major cause of harmful algal bloom. So a group with such ecological and economic importance, you think we would know a lot more about it, but actually in Singapore, well, before 2018, there were only about 20 species that were known, and a DNA analysis of just, we went around pulling plankton toes around, and we found 28 species. So we actually have more species than were originally known, just using those 29 plankton toes. That's phytoplankton. You have zooplankton, those small things that animals that are in the water column. Just 2 plankton toes, we found 10 new species records. All right. So a lot of unknown there. Not just the small things, but the small things that are living in other things, right? These are the symbiotic stuff, right? So of course, the most well-known are the Symbiodinaceae. These are what we call the zooxanthellae. These are single-cell organisms that live in corals, and that's what gives the corals the color, and is what makes food for the corals, and using sunlight, right? And of course, they don't just exist in corals, they are free living, but also they are living in other groups like giant clams and so on. Now, we used to think that, of course, they still look like this, right? Very, just green and blobs of cells and so on. But when you actually dig deeper and you do some sequencing on them, you find that they are actually very diverse. A lot of different types, or what we call species, of these single-cell organisms, and they exist in a community within corals as well as giant clams and a lot of other reef taxa. There's also a lot of temporal variability when we look at bleach or bleaching corals, right? So bleaching corals tend to lose the symbionts, and they tend to be dominated by one type of, or a few types of these symbionts. When they recover the diversity, and this is the diversity we talk about. Different colors represent different species. So it can be very dynamic, temporally and also spatially. Of course, we look at, again, a picture like this, you will find that things are hidden from us, not just because they're small or they are living in other organisms, but really because you have crevices and so on, which is actually becoming harder and harder to study. When I was a grad student, I was a student a long time ago, we used to go out, we were able to take large colonies of these corals, come back, and then we just crush the whole colony up. Because of the loss of a lot of the declining reef habitats, we are not able to do this kind of research anymore. But so we have to be creative to try and figure out what species are hidden, right? So what are the species hidden in these crevices, right? In between the corals, in between the other habitat forming organisms that we have on coral reefs, but also on mangroves, around mangrove roots and so on. Okay, so this is what we call cryptobiome. Crypto because it's hidden. And is a very rich area where we can find a lot of species. So what we do is because we can't bring back large numbers of colonies or corals, we don't want to kill them. Then what we can do is to sample them, is actually to deploy things that mimic the structural complexity of coral reefs. So this is something called the autonomous reef monitoring structure. It sounds more high-tech than it should be. Autonomous doesn't mean that it will move around by itself, but really it's just that we leave it there for 1 year or 2 years, and then at the end of the day, or at the end of 2 years, you find that they are encrusting with lots of organisms living on top and in between those plates, right? So this is deployed in Singapore. We did this over 2 years. A few years ago, we deployed 12 arms units over 2 years at 4 reefs in Singapore. Right, so at the end of 2 years, some of them look like this, some of them look even more encrusted with things. So without having to destroy reefs, we can recruit them onto these things and then bring them back. And what's good about this is they're standardized. We deploy the same things here as others deploy in Indonesia, in Hawaii, and then we can compare the biodiversity across these areas. So they look like this. You take them out of the water after 2 years, and then you dismantle those plates, and those plates, underneath those plates, you also find encrusting stuff, right? So it's pretty fascinating. And of course, these are just encrusting. We also get a lot of things that are crawling out of those plates as you are taking them out. And so you get a fish, you get also a lot of invertebrates, worms, and shells, and so on. Now, what's really interesting about this is that while it is a lot of these species are known, right? What we found is only 50% of all the fauna that were collected from these units had species level identification with some genetics, with some barcoding, which means that it's possible that we can get up to 50% of all the fauna that were collected from these being new species, right? Or up to 50%. What's also cool is you will find, there are a lot of species that recruit, almost 1,000 eukaryotic species. Now, in Singapore, there are about 4,000 known eukaryotic species, right? So we were able to recover just within, that those units are less than a square meter in area, right? And the total habitat that we actually sampled from those 12 units is just 10 square meters. So within the 10 square meters, you could get 1,000 eukaryotic species. That's pretty remarkable. That's a quarter of all the marine species that are known, or at least that recorded. Right, and you can see that what we call these accumulation curves are still going up. So we do need more ARMS units to ensure that the estimates are stabilizing. But there are also a lot of bacteria, more than 15,000 bacterial sequence variants, when we consider them bacterial species. And that is even worse in terms of the stabilization of the estimates. So clearly we need a lot more of these units, to deploy more of these to understand what is the actual richness. So how many do we need? The hidden stuff that we need to be found. If you can deploy more of this, so this if you project that number of units, so currently we're at 12, it probably take about 40 to 50 units to be deployed in order to get a good sense of how many species there are. And it turns out, I think maybe we will be able to maximize the diversity at about 40 to 50, recovering up to 1,000 new species when we get to that point. So we can expect to get up to 1,000 new species, we think. Yeah. Right, so those are the hidden stuff, okay? And perhaps what we can do about it. But also maybe we want to turn our attention to what we want to focus on, right? And especially in the marine environment. And this is something that you and I, we go out to the intertidal, we will see a lot of these, squishy blobs, and you will see lots of them. They look very similar to each other, and they just look blobs of things that you can't really identify. And those really need attention. So this is a group called Corallimorpharia. It's a sister group to corals. And they have never been studied in Singapore until a few years ago. And just looking at them, it's really hard to identify them. But if you look closely and you have used some genetic tools, you can we know that now there are actually about 6 species present in Singapore. So these squishy blobs could be corallimors, could be anemones, a lot of jellies, and a lot of things that, once it's above water, it just becomes very difficult to identify. And those are we need to turn our attention to them. And there's the plastic stuff. So what's plastic? Plastic is basically a term which suggests that organisms grow differently in different types of environment. One good example are corals. So corals, many species tend to be plastic, meaning that if you so this is actually the same species. In fact, they are the same colony. You fragment them into 2 colonies, you put one in a high flow environment and one in a low flow environment, they will turn out to look very different. In fact, they look like they are 2 different species, right? This is grown over a few months, and you can see that they look completely different, but actually they are from the same clone, right? They are actually genetically the same next to each other, right? So that is not, this is quite remarkable to be able to change the morphology of species. The other is just looking at one single colony of coral. One is on the top side, and this is actually on the bottom side of the colony. And you would actually, even for seasoned coral taxonomists, you would identify them as different things, but actually they are the same colony, right? So this is a classic problem for corals, and that's why corals are very difficult to identify. And it remains so because of how plastic they are, right? And in fact, we know very much about corals. So you look at the estimates that corals are actually one of the most well-studied, the most, in terms of the highest proportion of named species, right? So the bar in gray is really high. That suggests that most of the species have been discovered. But actually, some of them still, they are still missing from my inventory. This is actually a new species that is in Singapore. I'm currently describing it as a new species, right? So even these corals that you find at Hantu and so on, some Pulau Hantu, some of these reefs that you visit have new species, right? Despite them being some of the most highly frequently dived places, I would say frequent in the region. Okay. So other than the the plastic stuff, there's also cryptic, right? And cryptic is a bit different from cryptobiome. Cryptic in this sense means that they may represent different species. So these are actually two different species of oncydid slugs. They are genetically very distinct. And the only way to tell them apart is actually to look at their reproductive organs, right? And you need to do SEM and so on. So it's actually quite challenging to identify them. Also, crinoids, these are really hard to identify. These morpho species really differ in the ornamentations on the pinnus, and so these actually turns out to be three different species, even though we've not really been able to identify them properly. One of those characteristics, which really quite illustrates how difficult it is to identify them, is this structure called syzygy. These are perforations. You can't really see it now because it's covered by tissue. Now what you do is actually, if you what you will do if you were going to want to identify these species, is actually you will put bleach over it and dissolve the tissue, and this is what you can do, right? So you put bleach over it, and you can see that the dotted line appears suddenly. So this is actually a characteristic that is used to identify this genus. So without actually having, you just can't look at it under the microscope without, just looking at it, you will be able to tell what species it is. Right, and of course, apart from all those things that you can actually find, are also species that may have gone extinct. Okay, so we are in a biodiversity crisis, right? Species are being lost every day. And corals are also being lost every day from our local waters, for instance. And today we know that we have in this group of corals, the Poeciloporidae, or what we call cauliflower corals, we actually only have one species left, right? This is the only species in the family left, Poecilopora acuta. You can still find them in our waters, but all its other members of this family have gone extinct, right? So the first one, or rather the last one that has gone extinct, is Talophora pistillata that died in a 2010 bleaching. There were other species, Poecilopora vericosa, that haven't been observed since 2008. And this is quite interesting. This is a colony that came from 1950s, right? And the last time it was observed is 1960. And so actually, because it is past 50 years of its last observation, we only just recently declared this as extinct in the third Great Data Book. There's also another interesting one. This colony is found in the Smithsonian, and it was collected from Singapore, and it has never been observed since 1881, right? So that's pretty long time ago. But anyway, all these four species we presume may have gone extinct, leaving the only one. So if for corals, we have good record for corals. A lot of people are looking at it, a lot of people are studying them, but really not for other groups. So we don't have that kind of fortune for the other groups where you actually know that they have gone extinct, right? They would be silent extinctions, or we call silent extinctions, where they are gone without actually anyone knowing them. And finally, I think it's important to highlight, other than extinction of species, there's extinction of taxonomies. Not so much extinction, but just the inability of our numbers as taxonomists to keep up with the discovery rate that is needed to identify and to recognize species. So when you go out to the, just an example, you go out to a reef, right, anytime, your attention is always drawn to corals. You get, large fauna and things like that, seagrass. But actually the most dominant things here are seaweed, right? So these are actually what the groups that really require a lot more attention, okay? And what we need to figure out is what are these groups and what we need to focus our attention on, right? And this is a good example. This is an algae that we used to think is called Lobophora variegata. It's very common across all of our sites. But if you look very closely, it turns out that there are 4 species, and we only knew this 2 years ago, right, using a combination of genetic and morphological tools, right? So these are groups that, and this is basically a 4 times increase in diversity of the group, right? And this is just one of the species per group. So you get a lot of algae and animals that are poorly studied that we know very little about, and those are where you can find a lot of new species, a lot of new records. So we need to start this process of de-extinction of taxonomies. So as a side note, I just wanted to highlight, NUS is continually looking for bright students to join as graduate students. And so some of you who are interested in taxonomy should join us, right? And we have an info session at Department of Biological Sciences in next week, so feel free to join. And I think that's all I have. I'm happy to discuss further. I think I'm sure there are other things that I'm missing, and obviously there are lots of things missing. That's why we are talking about this, and hopefully we get more insights from the audience. Thank you so much for your attention. Over to you, Jasmine.
Thank you so much, Danwei. So we will proceed with the Q&A segment now. I think there are a couple of questions in the Q&A box and the chat box. So I will read out the question from the Q&A box first. So this is from Kawai. He asks, what is the arms unit made of? I guess the material.
I think they are fiberglass, if I'm not wrong. Are pretty hardy. And so they can't be burrowed in. Yeah, so things just encrust on it. Yeah, but so each unit is actually only just is this size, right? Quite tiny. In total, all the plates, if you put them together, it's only about 0.8 or 0.9 meters square. So it's a very, very tiny area. But the benefit is that you can carry it around, you can move it around, you can deploy it quite easily. They are relatively light underwater. But they account for a lot of these crevices that you may be difficult to find, difficult to dig into on a live reef, right? And the only way to do that really is to remove the whole colony, remove colonies and then study them. But ARMS is really helping us to recruit and then to also standardize across different areas that we are, studying. Yep.
One more question regarding arms. How feasible is it to deploy 40 to 50 arms units to study the maximum diversity? Are there ongoing research at the moment?
So yeah, good question. So actually we are we are going to have a new project in the museum, and we'll keep it a secret for now. It will be launched next year, hopefully next year. But part of that big project is to deploy these arms. Now, actually, it is not, well, because I wouldn't say easy to deploy, because we still need manpower, and I know my students and staff will complain, this is really hard to do. But actually, relatively speaking, deploying is not that difficult, is not too heavy. You can deploy with a 2-man team, and it takes maybe about 15 minutes to knock it in place on the reef, sort of a dead part of the reef. And so deploying 40 to 50 probably takes a month or so of hard work. But actually the harder work is at the end of it. So after 2 years, when we extract it, that's when you actually, each arm actually takes around 2-3 hours to dismantle and to process. So you actually need to scrape out all the organisms. You need to pick out the larger stuff, and then you need to blend it, and you need to sieve out, sieve in different size fractions, and so there's a lot of work that goes into processing the arms, right? About 2 to 3 hours per arm, and that sometimes takes. So if you remove 10 arms, that is like a 24-hour operation that actually takes a lot of time, and that's just processing it. We are not even thinking about sorting them yet, so we have to sort them. Often we use genetic tools to help us. So the whole project, the 12 arms project that we did took us, I guess, about 4 years in total to get it to completion, right? So deploying it is sort of the easy part. Removing it is also not that difficult. It is the processing and also sequencing analysis that really it makes up bulk of the the effort. Yeah. So it is feasible. We need a lot of help, and of course we hope to involve volunteers during our project.
Okay, this question is from the chat box. I think the participant here is asking this question in reference to the slide on the plastic corals. So she asks, if they are the same species, why do they have different names? Is it because they were believed to be different species before a genetic ID was done?
In a way, yes. So when you are a taxonomist, you go out and collect, right? And you go out and identify corals. You don't have the history of how they grew. So maybe a coral could be one single colony and it broke into two, and then somehow they just ended up in two different environments, and then they grow differently. So over time, what you find is that you have all these a mosaic of corals that some are the same, some are different, and they have different growth histories. When you are studying corals or any group, you go out there, you have to make certain assumptions, and those colonies that look different, you call them different things, but you didn't know that they may have had the same genetic origins. So those are the challenges of without genetic tools. Those are the challenges, and that's why there has been in many groups some confusion with names, with taxonomy. I won't say corals is the only one. There are most other groups. They are also the same. You get this change in names and so on, which a lot of people complain about. But it is part of the process of trying to understand diversity. And of course, it's more inexcusable if, let's say, it's one colony and then you look at one side of the colony and the other side of the colony, they look different. You should, of course, call them the same thing. But the example that I showed is just to illustrate that, if you don't look closely enough, you might mistaken them for the same species or different species erroneously. So it's very important to be very precise and very detailed when we look at species. Yeah, so that's the science of taxonomy, and I think that's really important.
Okay. One question from one of the participants: do you study marine isopods?
No, well, I mean, I would say no. No is the answer, because there are experts out there who study isopods. But we have done some. We had a big population genetic project where we collected around 12 species of marine organisms, and the sea lice, we call it sea lice, the sea cockroach that's crawling around seawalls. We did use them as one of our model organisms or one of our study organisms. We actually found that there are two very good species in Singapore, but they really look quite indistinguishable. And what's interesting is if you go to some of these seawalls, even within the same island, you have one seawall that only has one species, and you walk 5 meters to the next seawall where there's a broken part, where in the same island, 5 meters away, that seawall has a different species altogether, and nobody was able to identify them, except unless you use genetic tools. So that illustrates how difficult it is to identify species without help from genetics. And of course, a lot of us are just amateurs, right? We're just going out and looking, me included. So we do need help from a lot of these modern tools that we can get. Of course, eventually we will need to involve the experts, the taxonomists who work on these groups in order for them to make sense of these differences. Yeah, thanks for the question.
Yeah, we have a fresh question coming in from Won Lake. How many new coral and other marine animals have been discovered in this project?
So this is I guess I'm assuming you're asking about the ARMS project. We think this is just back of the envelope calculation. So we have about 1,000 species. There are not that many corals on those things, and they probably just recruits that recruit on them, and we scrape them off. So there's no way we can actually describe them. There are some larger things which we'll remove as wholes, and those we will need experts to come in and verify. But just looking at the number of unknown things that we sequence and we will not be able to match anything on the public database, it looks like there are at least 100 new species across those 12 arms. And it's just very difficult if we don't have the organism, the whole organism it's really hard to actually describe them. So the next stage of this project, if we deploy more, is to be very careful and to actually remove things more carefully so that we are capturing entire organisms. And a lot of them are just encrusting. So that means they are like flat on the plates. They are very difficult to identify. So we need to involve experts right at the start. So when they're helping us to process, they can then maybe do some preliminary processing and sorting so that we can fix them or can preserve them correctly. Yeah. So what we do now in the previous project was just scrape everything and then we sequence everything, right? So it's not really meant for good taxonomic work, right? So yes, we discovered them, but it's almost impossible to name them properly. Yeah.
Okay, a few more questions. This is from Sebastian. What environmental factors would influence biodiversity between the different locations that you sampled?
Right. So this again, assuming it's the ARMS project, I think there's this assumption that in Singapore, because it's a very small area, the environmental factors, parameters and conditions are quite similar among sites. I think that's generally true if you look at, of course, you compare Singapore with a temperate area or compare it with a larger area, Australia, for instance. That is probably true, but there are actually micro environmental factors that can influence the biodiversity, right? And so where you find species is also quite specific. So if you go to a very, go to certain sites, you can just within a site deploy arms, those units, one on a reef crest, right, or one at the reef slope. You can deploy them under other corals. You get very different things growing, right, over 2 years or 3 years. So there are these what we call microenvironmental variations, and that the differences may be small in each of those parameters. So when we look at light, it's very different, but very different in very small ways. But if you combine all those parameters together, light, temperature, exposure to wave action, all that together, you can get very different environments, even within a small area, let's say about 10 square meters. You can get very varied environments. And that's what we're focusing on. We're trying to understand what are the microenvironmental parameters that drive biodiversity at a small scale. That's an interesting question, and to me at least, because well, we don't have a choice. Singapore is small, so that's one of the interesting questions that you can look at. But of course, you can look at larger gradients, right? Differences between temperate zones and tropical areas and so on. Yeah, but there are factors that separate many of these locations, even in Singapore.
Okay, I will combine the next two questions. Are there coral species only found in Singapore? And what is, in your opinion, an area that needs immediate research attention given the limited resource?
Okay, so this is a good question. All right, so all species, you think about it, right? So all new species, when they are discovered, let's say they are only discovered at one location, then it's endemic to that space, right? Initially when you discover them. Of course, then later on they get found in other places. So happens that this particular coral that we are trying to describe, I think we found it in Raffles Lighthouse. This coral is only so far known in Singapore, right? So it's the only endemic species to Singapore at this moment, but I'm very sure we will find it elsewhere, right? So Singapore is very small, and our corals, they are very well connected with reefs from Indonesia and also from Malaysia, and also the broader South China Sea region. So we share all the species with the region, right? Except for this species, because this species has not been described. So when we describe it, it will be endemic to Singapore, but only for a short while. So after someone finds it in Indonesia or Malaysia, then it's not endemic anymore. Yeah. So in my opinion, what is the one area that needs immediate research attention? I think really if you just drill down to try and understand biodiversity very, very broadly. So what lives there, then I think would be the smallest things. And I just give you an example. The smallest things can have very big effects, right? So there's this genus of bacteria called Prochlorococcus, and this genus was only discovered about 30 years ago. So we've known oceans for centuries, but we only discovered this species of bacteria only 30 years ago. And it turns out this group of bacteria accounts for 20% of the oxygen production in the world, right? One of the most abundant species of bacteria in the world, right? So if you ask me, I think if we focus our efforts on the small little things, they can often have very big effects because small things tend to be abundant, right? And when they're abundant, they have a very big effect on the environment. So I think that would be the priority, I think, for biodiversity discovery. Of course, when we talk about bacteria, it is a whole set of experts, right? So none of us in the museum, for instance, we on bacteria. So we need microbiologists to be in the picture. We need even cell biologists and molecular biologists to join in this effort.
Okay. The next question, do you see tourism as a possible way to engage the public in marine conservation? If so, how?
Yeah, it's a good question. It's a question that has, a lot of conservationists have asked, right? And ecotourism is one growing business industry, and it is very well established in many parts of the world, right? So if you go to places like Maldives or very, very good dive sites, often there is a team of marine biologists who are there to as outreach. They are like our outreach and education unit, but for hotels and resorts and so on. And I think if they are well trained and they know the environment very well, they focus on the right things, the right messaging, I think it's possible, right, to engage the public, to get the public interested in conservation in the right ways, right? And what you don't want happening is that maybe they give out the wrong message, right? A wrong message would be, I know of this example where a resort, they sort of they bring lots of divers down, right? And so they don't have the right ratio of guides to tourists, and then the activity they were doing is actually to transplant corals. So what do? They go to the live reef, they pick up corals, they take out branches from the live reef, and then they go and transplant it somewhere else. So that's definitely a wrong way to do it. But you can see how it can the message can be, you can make mistakes in that way lah. And so I think as long as the people who are doing it, they're well trained, they know what they're doing, I think that's a good way to enthuse the public in conservation. Yeah, so that's how I think, in general, I think it's possible for sure.
Okay, I think with the time we have left, we will just answer one last question. This is from Roseanne. Why do you think taxonomies are going extinct, and how do you think we can get government or private donors to fund taxonomies?
This is a century-old question. I don't think they're going extinct. I think there are more taxonomies for sure, right? The fact that we have a museum and half of our staff are taxonomies, I think it's testament to the increased attention that biodiversity is getting. But I think I want to put it in context is our goal is to try and describe all of the diversity on Earth, right? So and that's 10 times of what we know today. The rate that we are going is much slower than is needed. So we need a lot more, right? So I don't think taxonomies are going extinct. I think we're a growing group of people. But getting government or private donors to fund is always a challenge, especially government to fund is a challenge. A lot of private donations have come in. As you know, the museum was built with a lot of public and private donations, and we're very grateful that actually a lot of philanthropists are very interested to conserve biodiversity and to see biodiversity being documented and to educate the broader public on what's there and what their functions and how all of us can be part of this conversation to conserve species. So I think just funding taxonomy, I think you will be able to get small bits of money, but I think we need to look at this in a broader picture in that taxonomy is not just about describing species, but it's describing species so that eventually we get the goal of understanding what's there and what the functions are and what do we need to conserve, right? So getting from the basic science to conservation is really important, and we all need to have that perspective. Otherwise, yes, it's important to describe species, but we are doing that for a purpose, right? We're doing that to try and understand global biodiversity, and so that becomes then our work becomes more urgent, and hopefully that attracts more funding.
Okay, we have a very interesting question in the chat box. Do you
Huh?
have a favorite coral species? If yes, why?
Oh yeah. My favorite coral species is not really species, but a group. If you go out to the reef, right, and you just look around, you will see these massive corals. And the reason why that's my favorite is because I started working on those when I was a grad student. And it's the most abundant, one of the more abundant groups in Singapore. And they're very hardy, right? So they sort of embody what the reefs in Singapore are, right? They are actually all over the place. They are very hardy, they're very resilient. And they're not very pretty, right? So if you go to the reef, it's not like conventional. You go to the Great Barrier Reef, it's a very nice reef, but they have their own beauty, right? So they're boulder shaped, and they come in all sorts of sizes, lots of colors and so on. So I think that's why I like. So there's a group called marulinids, right? So most of the boulder corals are like that. Yeah, so that's my favorite coral group.
Okay, so thank you, Danwei. I think we are approaching 9 p.m. So unfortunately, we will have to end the Q&A segment here. So thank you so much for giving us such a detailed explanation and overview on marine biodiversity exploration, especially in Singapore. So before we end off today's session, let me just quickly share my slides again. So I would like to request for everyone to fill in the feedback form for today's session, and I will forward the responses to you, Danwei. And also, while you are filling up the feedback form, let me just quickly put the link in the chat box first, the feedback form link. So while you are filling up the feedback form, I just like to quickly advertise for our next talk shop session. So on November 1st, we have a guest from NTU. So the guest speaker for our session on 1st November is Mr. Clement Ng, a PhD student at the Asian School of Environment, Nanyang Technological University, and he will speak to us about the ear stones of fishes, which is interestingly they form growth rings as well, just like trees, and they can inform scientists on a wealth of information such as the age of fish, their migration patterns, and so on. So Clement will be speaking to us and explaining how fish ear bones may actually hold the secrets to help us develop tools to better manage our seafood supply as well. So the publicity for this talk session is not out on our social media platforms yet, but if you are interested and would like to sign up for the session now, the registration link is already live, so I will put in the link in the chat box as well. Yeah, so with that, we will end our talk session here, and I hope everyone has a restful night. Thank you so much.